e con la professoressa Lavinia Tagliabue che è adesso in visita nel laboratorio di Marco Agus e aspettiamo ancora Lina Ibrahim che si connetta per cominciare la nostra conferenza. Marco, sei connesso? Che non ti sento. Also colleagues from Qatar, from Saudi Arabia, are we here? Because I see that we have a KVL connected, but we cannot hear your audio. Hello. Hi. Okay, now we can hear you.
Hello. Sorry, the controllers just stopped working.
Oh, okay. That happens.
So Lina's just calling the audience.
Okay, yeah. We can hear you, so that's good. We have already. Okay, we have Marco from Qatar.
Yeah, Marco's audio is lagging. Can you hear me?
Yes, the audio, Marco's audio is a bit lagging, yes, unfortunately. Yes, Marco, we cannot hear you basically. But sure we could hear him very well. And is Lina connected?
Yes, I'm here. Hi, Corrado.
Oh, it's ADL?
I can't do properly.
And Marco, we cannot hear you. Yeah, the
audio from Marco is really bad, unfortunately. Marco, can you mute yourself and then unmute yourself? And then, I mean, maybe we can we can just try to connect you later, Marco. Okay? Okay, we have a nice crowd. Can you hear me now? And Lina. Yes, Marco, now we can hear you.
Okay, I cannot hear you so good, but there is some setting for the audio.
No, it shouldn't. I mean,
It's
fine.
okay, it's
Maybe
okay, I
it's
can
your
manage
network.
it.
Okay, wonderful. Then I think we can start. So thank you everyone for being here, either in presence or remotely. As you can see, we are here in this room, which is a sort of avatar of the Institute of Anatomy of the University of Turin. And today it starts the Brain Awareness Week of Saudi Arabia. So to honor this week, we have organized an event together with KAUST and HBKU in Qatar, where we will talk a little bit about brain research, and how we can use this technology, virtual reality and metaverse, to do research, analysis, and eventually also work in classroom. So I'm honored today to introduce our first speaker, Lina Ibrahim, who is an assistant professor at the BSc division in KAUST, who is going to start to talk to us about her research in neuroscience. Lina, the stage is yours.
All right, I just need to figure out how to navigate my slides. Just give me a second. All right, thank you everyone for joining virtually in this metaverse. So this is obviously a very exciting time for, in terms of technology and how different scientists can actually interact with each other no matter what the geographic location is. So I'm just going to give a very, very broad overview of why it is important to study the brain in this level of detail, right? So, so let's get started. All right, so we know that the brain is a very, very complex organ, right? And this is an example over here in this 3D environment, 3D reconstructed brain from the University of Torino, where you can actually see this half a hemisphere of the brain, which is also represented here in this diagram, right? So the brain is actually this very in a complex connection of many different cells connected to each other, right? And all of the brain functions that emerge are as a result of this complexity. So this is another diagram to show how all of these different brain regions connect to each other. And there are obviously a lot of different regions of the brain that are important for many different types of functions, right? So if I zoom into this, you have the visual cortex in the back. This is the part of the brain that controls visual information processing. You have the motor cortex, which is important for getting all the information into this motor area and having a specific behavioral output, or a specific motor output rather. And then you have your prefrontal areas, your language areas, and so on, right? So this whole brain is basically divided into these functional categories, but we need to understand what's actually inside of each of them, right? How are the neurons within each of these brain regions? How are they, what are their shapes, what are their electrophysiological properties, how are they connected to each other, in order for us to really understand what's going on. So if we want to decipher brain circuits, let's say you're taking a very simple behavior, like reading a book, right? So your the whole book is composed of many sentences, many words, many letters, right? So the behavior of reading a book can actually be broken down into several different aspects, right? So you have the pathways, you have the local circuits, you have at the level of the single cell, right, at the level of molecules and the level of genes, right? So all of these different aspects is required and integrated with each other in very precise ways in order for us to be able to have a specific type of behavior, such as reading a circuit. So in order for us to understand this behavior, we need to understand what happens in the brain at the level of individual neurons, right? And this is basically an example of a few neurons that are reconstructed in the mouse brain, right? So each of these different colors is an individual neuron, right, with all of its processes, right? And it's very important to really get this level of detail because we need to understand where the signal goes, how the signal is integrated, and then eventually what actually happens, right? So neurons in different brain regions, they have very different morphologies. These are some neuronal reconstructions from Santiago Ramon y Cajal. So you can see a Purkinje neuron with all of its diverse projections. You have pyramidal neurons in the cortex and retinal ganglion cells in the retina. And you can see how different all of these different neurons are. And these neurons have to actually be different because they're in different locations of the brain, but also the fact that they have to perform different types of functions. So they have different, very, very different properties, right? And all of these different neurons with different shapes and sizes have a lot of different electrophysiological properties as well that allow them to function the way they are supposed to function. And for us to really understand the complexity of the brain and the complex processing, we actually have to understand the brain at the level of the whole brain, at the level of its connectivity, at the level of the local circuit, at the level of its synaptic inputs, which you can see over here, and at the level of the EM, so at the electron microscopy level, how these different synapses are actually connected, how they are formed, and how different neurons connect to each other. In order for us to eventually be able to understand and put all of these different things together, form a single neuron, to have a specific core circuit motif to a larger scale architecture plan, which in the case of the analogy of reading is the sentence, and then at the level of the whole brain. And so the structure is very intricately linked to function. So we have to understand the structure of neurons, the structure of the brain. And this is actually an example of one of those neurons, right, with all of its morphologies. So you can see how complex it is, and imagine that you actually have 80 to 100 billion neurons in the human brain that have all of these different morphologies that have to connect with each other in such precise ways to be able to have a specific function. Sorry, my VR is a bit messed up. All right. So yeah, that's it for me. I think Corrado is going to take over now, who's going to talk about how do we actually get to this level to be able to segment these neurons in such a nice way and really try to understand what's going on in terms of the very, very details of the microstructure of at the level of the individual neurons. So Corrado, you can proceed.
I don't know, can you hear me? I cannot hear Corrado.
I am here, sorry.
Okay.
I was just giving some visuals to the rest of the audience, as you might see. So I was running back and forth.
You were saying that you were setting up the device for other people.
Okay, very well. So thanks, Lina, for your talk and for showing us how we can study the brain at different levels. So now I would like to talk a bit more in detail of how we can study the brain a bit more in detail using other techniques and using other methods like electron microscopy and 3D reconstructions to analyze the structure of the brain, which is something that basically is my almost everyday job, right? So what Rina has shown you here on top is a neuron. And in particular, this is a 3D representation of a neuron. In order to represent this neuron in 3D in such detail, it is important to image it with a microscopy technique that allows us to see it at its finest details. I'm sure that you have all seen at least once a microscope. Microscopes are objects that can magnify things that are very, very small, like cells, of course. But like everything in physics, microscopes might have problems in deciphering some types of shapes, and depending on the fact that different types of microscopy techniques might be limited in resolution. So we might really not be able to see very, very small objects. And this is important when we talk about neurons, because a lot of times what we are interested in are very small details like synapses. And synapses has a size which is in the order of hundreds of nanometers, which is too small to see with light microscopy, and everything that is located inside synapses. So in order to overcome these limits, what we can do is that we can image the brain using electron microscopy. The image that you see here is an image of what we call the neuropil. The neuropil is a part of the brain that contains just connections, and here is a list of everything that you can see under electron microscopy. This is a part of the class that I give to my students. Anyways, here we have dendrites, we have dendritic spines, we have boutons, like this object here. This is a synapse. A synapse is something that is very, very small, of course, and we cannot image using light microscopy. So, electron microscopy allows us, because it has a resolution of in the order of nanometers, to visualize this very small object. Thing that is important about electron microscopy is that together with synapses, another thing that allows us to see is associative processes. Astrocytes are the brain. Astrocytes are cells like this one. Let me
grab an astrocyte. Okay. I hope that you can see it. So this other cell here is an astrocyte that looks like a bush, is a bit more complex in shape, and has all these very little small processes. These small processes here are located at the interface with synapses, and their role is very important because next to synapses, it allows these cells to provide energy. Hence to control the energy management of the neurons. And in the neurons, the synaptic connections happens at these very small tips here that are called dendritic spines. We have billions of them, and at these sites it is possible for other neurons to create connections. Now, dendritic spines, together with boutons, create these very fine structures that are called synapses, and I can show you how one synapse looks like. This here is indeed a synapse. So these are very small processes. This whole trunk has a size of 5 micrometers, and here between this blue structure, which is called the button, and this other green protrusion, we have a sort of patch, which is the synaptic connection that contains channels that allows the neurotransmission between the presynaptic and the postsynaptic terminal. Of course, bidimensionally they look very different, but three-dimensionally we have these very complex structures and these very complex shapes that we can study in their finest detail, because the change of their shape has a functional meaning. When they change, they can basically modify the connectivity and enhance connectivity that are enhance connectivities that has to be loose, and power connectivity that needs to be strengthened, and this is the basis of learning and memory. So in order to visualize these nice three-dimensional structures, and this is something that then our friend and colleague Marco Agus will talk a bit more in detail, what I do is that I image these structures in 3D using electron microscopy. Like in this case, this is a cube of tissue of 100 by 100 by 75 micrometers that I have used to reconstruct, for instance, this astrocyte here. This is a fairly large field of view in terms of electron microscopy, while for this synapse is much smaller. And then we recognize individual cells based on their morphology. Like this cell in blue here is a neuron, this cell in green is an astrocyte, and these are other type of cells, microglial cells, which is a sort of immune system of the brain, and endothelial and endothelial cells and pericytes. And then we can reconstruct these structures here in 3D. And I can show you how these cells are intermingled together. So this is a reconstruction, the same that you are seeing in the picture in the slide, with the blood vessel here, an astrocyte in green, and a neuron in gray. And by studying how these cells interact morphologically, we can infer their physiological type of functioning. Now you understand very well that being able to analyze these complex morphologies in 3D is not that trivial. And this is why we collaborate very tightly with Professor Agus, who is an expert in visualization, in the scientific visualization, in order to extract qualitative and quantitative parameters from these three-dimensional reconstruction for the types of information that we need. And in particular, together we have explored the properties of energy management of astrocytes to synapses in these small cubes. So saying that, I will leave the word to Marco. So Marco, the stage is yours if you want to take over. And thank you everyone.
Can you hear me?
Yes, we can.
Yes, we can.
And you know, I will more or less introduce what I do for these guys. And actually, you know, I'm one of the guys that start from the image that are extracted from the microscope and try to provide the tools for Corrado and also Lina that help them for the analysis and for getting insights. So, I don't know, yeah. And normally whenever I present my job, I like to show one slide.
You want to make it bigger, Marco?
Yeah.
This
I'd
one?
like to show one slide, in which I'll show how the data can be represented with a different level of abstraction. And I normally try to compare the data representation that we always create to modern and to historical art. So you see from the very left painting from Caravaggio on the top, and on the bottom the traditional way that scientists use for annotating the inverse image. They normally indicate what are the important features in the image, and they put some sort of annotation to drive the narrative and to explain what is happening. After that, since the development of processing resources and also the development of GPUs and so, people started to be able to perform and to create 3D reconstructions like the ones that you are experiencing right now. And some various methods they can be used for comparing the real stack with the 3D reconstruction, for performing visual analysis and to cut the areas that they are not of interest, to make objects appear and disappear and so on. And this is part of our work to create tools that help them to interact with objects and to have a clear 3D idea of how the objects look like, as the same way that you are experiencing right now in the metaverse. On top of that, what we are trying to do is to highlight important portions of the data and also to incorporate the outcomes of simulations. So you can see in the first pictures that you can see here we try to incorporate the outcome of energetic simulation in a way to understand which part of the neuron absorb more energy, where this energy is coming from, and so on. And then a part of that, what we are trying to do is try to abstract all this kind of information in a way to create some sort of image that they are containing more or less all information together in a very concise way, as like the picture that you see on the far right, that is associated to the Pollock painting. In this case, we have a very abstract representation that is very difficult to understand for layman people, and sometimes even for neuroscientists, but with training and with experience, they are able to get insight even from this kind of picture. So, as you can see, our job is starting from the real data and is getting into creating some sort of very concise visual representation that they can provide insight to the scientist. Now, I don't know, I need to switch. Let's see, it works. Okay, here is one example. And then what we did in past also together with Corrado is to create virtual reality tools for performing immersive exploration. Tools that they can provide, for example, a skeletonized representation and tools also for performing interactive analysis and so on. And on top of this kind of study, what we did is to try to explore the metaverse ecosystem. And this is actually what you are seeing right now, the outcome of the main efforts of putting together the various kind of data sets that we created, and also the outcomes of the design process where some collaborators created this really nice environment where people can have discussion and can collaborate each other. We already showcased this kind of environment in other events, and also we presented in a conference in Portugal last year. And actually we are even expanding this this part by trying to incorporate even more modern technologies, like, for example, one example of what we are trying to do. Here, put in here. These are the first experiments in which we are trying to train large language models on the neuroscience literature, and we are trying to create deepfake of neuroscientist experts that they answer. I don't know if I can start the video. Yeah. I don't know if you can hear the voice. The guy speaks English much better than me. And these are the first experiments that we are doing of deepfake technologies associated to this domain knowledge, and this deepfake expert is supposed to be a virtual agent that can help users in exploring the 3D models and answer the questions related to this object. And apart of that, I think that another topic of interest for us is to help scientists in the segmentation of data, so in the automatization of the creation of the 3D models. And apart of that, I think there is I'm okay, and I'm done with my contribution, and I'm here to answer to all your curiosities and to all your questions.
Okay, thank you very much, Marco, and thank you very much, Lina, for your intervention and for making
this conference, which is also a little experiment for us, because I mean we have to think that we are live with our avatar between 3 continents, and somehow we managed to share this room with many people. I mean, here we are at least 4 people connected simultaneously. You can see from the live stream of the photo booth that there are other people in the people in our room. Someone that you might recognize are my father and my mother, and I think that we can, I mean, if there is someone that wants to try to enter into the metaverse room, I have other VR headsets here locally that, but I have to do it myself with the other people. The room will stay open, so if someone from Coast side or from Qatar side wants to play around with the 3D models, I think that we have described what we have, and I want to thank you again for your help, and I hope this was interesting. And again, thank you very much for agreeing on giving us your time together. Okay.
You can go closer to our avatar
Marco, your audio is very bad for some reason. Yeah, okay, we cannot hear you very well, Marco.
Now can you hear me?
I hear you in high-pitched voice. I think it's a bandwidth problem, Marco.
Okay. Okay. Oh, okay. There are kind of people and there's something hanging from the ceiling. And do I walk through the table? Grab, okay. But I have to be near an object to grab it. Oh. Oh, I can bring it near me. There we go. Now I've brought a blood vessel and a astrocyte close to me. Got it. Got it. Oh dear. Christ. How do I do that? Zoom in. Oh my God. Oh, good heavens. And I can... Ooh. This is incredible. Okay, this is too much. There's somebody else right in front of me. Shoo, go away. Person with beak. Yeah, oh my God. This show I watched called Upload, it's a little bit like this. Come near me, you. Okay. Oh my goodness. No, I didn't. I wish I had. I had this giant skull in front of me. This astrocyte round specimen is amazing. I mean, this is just fabulous. So if somebody else moves it, it won't move. Okay, this is too much. Shoot. This is a little bit like, you know, spending hours of one's life in inception. Oh my God. Okay. I'll let somebody else have a go. I'll let somebody else.
Where are my hands? Okay. Okay. Yes. Okay. Okay. Okay, now see myself there. Okay, and what ah.
Ah sì, giusto, sì, sì.
Ok.
Dovresti riuscire a girarlo sugli assi. Ah sì? Solo qui muovere come ci voglio.
Okay, let us see that. Hello. Yeah, this, but I'm moving with this one for some reason. Okay. So it's okay. 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 Much better. Okay. for moving, and this for grab, for grab. Okay. Okay, perfect. Okay. What is this? Interesting. Nice. Very nice. Wow. It's nice. Very nice. Right.
Meglio.
Okay. Yeah, I think it's fine. Yeah. Oh. No, this is interesting. Uh-huh. Where did it come from?
change it.
What?
Una delle porte, se vedi due porte, in una sopra c'è scritto sala settoria, se vai dentro quella porta entri nella sala settoria.
Devi soltanto avvicinarti, c'è lo posso alla destra.
Lo bianco lo rimane alla velocità. Ok. Sì, sì, lo giusto. Una volta in Giappone che lo store della Apple lì erano già in esposizione l'anno scorso, c'era dentro
erano i cosi di acquavigio.
Sì.
Io non li ho mai provati.
Però lì non ci sono controller, ci sono un sistema di
Sì.
che vedono anche i tuoi movimenti e quindi loro lo eliminano quel pezzo di algoritmo.
Sì, ma è come il Microsoft HoloLens.
Non conosco quelli. E credo non siamo mai arrivati in Italia per motivi di scarsità proprio dei approvvigionamenti di pezzi, penso.
No, in realtà sono studiato, però dovrei comprarli un paio. Sì, per un altro progetto. Però non so quanto magari costano comunque parecchio, sono 3.500 a volta. Che
I'm not getting the image.
di Microsoft HoloLens.
Oh, you can see you.
Cioè, non
Something is wrong. Yeah, yeah.
so giudicare quanto sia
Literally, yeah.
tanto più potenti.
5.
Questi per me sono fantastici.
Sì,
Questi qua.
quello il tuo personale.
Something is wrong. And like, I'm not getting into the environment right now.
Diverso.
Maybe I have to get here. No, nothing. I can see all of you.
Che però hanno
I can see you guys actually. Ah, no, no, no. No, 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 it's fine, it's fine.
ha delle caratteristiche più avanzate, c'è l'eye tracking eccetera, però per alla fine per quello che lo vendono, che per videogiochi eccetera non serve tanto, quindi
Cioè è un po' troppo high end.
Yes.
esatto.
Ho capito. È un po' il discorso che fanno anche con il video. Dicono che la prossima tornata è un po' più low cost.
Sì, perché sennò che ci fai?
Tipo senza vetro, perché loro hanno questa funzione che puoi vedere
Sì,
anche
sì,
quello che
sì, sì, pass through.
però sono tipo di 1.000 euro il prezzo per quella roba lì.
No, non lo so.
Però affascinante.
I can see both.
Ok. Allora adesso vedo noi.
Wow, that's Can you see like each other very special? Where are you? like around which thing? I am around the skeleton. The skeleton behind around the board. Oh.
È strano, vero?
C'è un tavolo. Bene. Ok, con questo io concluderei la diretta. Grazie mille a tutti, ai colleghi in Arabia Saudita, ai colleghi in Qatar, quindi KAUST, HBKU, grazie all'Università di Torino che ci ha consentito appunto di realizzare questo ambiente e i mezzi per navigarlo con i visori, grazie al fantastico aiuto del team di RKH che ci ha prestato gli spazi e il supporto tecnico, che è stato fantastico oggi, nonostante le difficoltà tecniche di questo setup. È andato molto, molto bene, e grazie a tutti voi che avete avuto la pazienza di seguirci, se ci avete seguito, e spero di poter replicare altri eventi di questo genere. Buona domenica a tutti!